Now I would like to invite Professor Jayati Sharma ma'am, Professor Director of ASCO Department, Amity University, Rajasthan. Ma'am, please welcome our invited key speakers, Shandar Ahmed sir.
Very good afternoon. Am I audible to you all?
Yes. Absolutely. Loud and clear, ma'am.
Sorry.
You are absolutely audible, ma'am.
Oh, thank you. Thank you so much. Very good afternoon to everybody present over here, and a very warm welcome to our chair of the session, Professor Shandar Ahmed. I think we had a very brief interaction in the last conference also, and I have a good fortune to welcome you in this session. Thank you so much, sir, for joining us for this session. And I'm sure your presence would add more value to the research work that is going to be presented in this session. This is the 7th session and 2nd day. And a lot of good papers have been presented yesterday and since morning today. And I look forward to hear all the presentations today in this session. So a very warm welcome to all the paper presenters. Thank you so much for participating and adding value to this conference. And I hope that together we could make some difference through the theme of this conference, which is green environment and innovation. I will not take much time. It's the session where the academic deliberations are supposed to be done. So it's just a formality, but on that note, on behalf of entire Amity University, Rajasthan, I welcome Professor Shandar Ahmed and all the participants and look forward to very brilliant presentations. Thank you so much.
Thank you very much. Thank you. So should I start?
Yes, I think we can go ahead with the
Yes, sir.
proceedings.
Okay. Go ahead.
Okay. I have to share my screen. Can I do that?
So your screen was visible. Can you share it again?
No, that was not my screen. I think that was from your side.
Oh, yes.
Ma'am, I shared it actually.
Okay. All right.
Okay. So can you see my slides now?
Yes, sir.
Can you see full screen? Because I don't
Not in full screen, sir.
I think so the right window is not shared, so I have to change that. It should work now, I think. Is it there?
Yes, sir.
Can you see my full screen now?
Yes, sir.
Okay, thank you. So first of all, thank you very much to Professor Jayati Sharma and Professor Manju and everybody here, and Dr. Yudhvir Singh, whoever I know and whoever I may have missed, for having me here. It is a great occasion to be with Amity people again. And it looks like we know each other for a long time. And since this conference is about green technology, and I was perhaps picked up to talk about more of the innovation part of it. But after I discussed my topic, I thought to talk about how the biocomputing, bioinformatics, my area of research is now helping the development of green technologies. So the topic today is AI in bioinformatics and biocomputing, and how it is impacting how the computer-based experimentation actually is helping us to save environment. And in this, I'll start with a very something that is of interest to everybody. We are living longer today, and our life expectancy not long ago was very low. So even if we look for say the data for last few decades, we have been making rapid strides in terms of life expectancy. From over the period of 1,121 years, our life expectancy has increased from less than 30 years, just so shocking to know. In 1900 and before, our average life expectancy was less than 30 years. And even in 1960s, our life expectancy was close to 40 years. And in 2021, our Indian life expectancy is 60, more than 67.2, something like that. And if you look at other nations, we still have to catch up with them, some of the developed countries. And this graph is constantly growing. You will see a slight dip in 2021 because of the COVID. But in general, we have been constantly improving on the life expectancy. So on another side, more people, more food, and we also growing more food. For instance, the Indian data on the crop production is that we are in 1950-51 financial year, we were just producing about 70 million ton of say these pulses, coarse cereals, wheat, rice, and today we are almost 4 times of that. So constantly we are improving in terms of our healthcare and in terms of the food production and the agricultural produce. So what is driving this growth? The growth is being driven by longer life and greater food production is being driven by for the health. We have better healthcare, we have hygienic conditions, better water access, infrastructure, and medical sciences have grown fast. And the disease prevention, we are able to prevent diseases and overcome disease. Even pandemics like COVID, we were able to overcome rapidly with much less loss than what used to happen in the past. And better treatments, we have better drugs, we have better interventions, the surgical treatments, therapeutics have improved. On the other hand, the food production is because because there are more people, it is a compulsion that we should grow more food. So there is a greater farmland, we have better irrigation facilities, we have different varieties of our crops, which can now work in drought resistant scenario. And they are meeting our need for more food. I'll focus more on the first aspect. That is what we are doing in the field of the healthcare and the research in the field of bioinformatics and biocomputing with the help of AI, and how the field is improving over time. But now, first thing that we notice in this green and green technologies that we are talking about today, what are we doing? Drug discovery comes at a cost to environment and ethics. In this talk, I'm treating environment and ethics very, very closely to each other. So we have a lot of this research is being done on animal models. We are also spending a lot of laboratory consumables, which are toxic. We are sending them to the water streams. We are not really disposing them in the best possible way. And even if we are disposing them, we are filling our earth with a lot of consumables, wasted test tubes, wasted chemicals, and so many different products. So laboratory consumables, a lot of them we are using. And we are also doing a lot of clinical trials. We are subjecting a lot of people to diseases, clinical trials, and even before clinical trials, we are doing various kinds of research. So these are actually driving our longevity drug discovery process. So in some sense, they're indispensable. But to look at what is the cost we are paying, and this is the data taken from PETA, animal models for biomedical research. So this is just the data from US, and the data in India will not be very different. Every year, more than 110 million animals, including mice, frogs, dogs, rabbits, monkeys, fish, and birds, are being killed in laboratories. For doing this research, we are not producing that many drugs every year, but we are killing 110 million animals. Probably these 110 animals, a million animals, will be saving lives of a few thousand people in the long run. And irony is that 95% of all new drugs that are shown to be safe and effective in these animal models actually fail in human trials. So basically, we are killing about 95 animals for just making 5 successful experiments. So many of these scientific experiments also involve painful treatment. So when we use animals, what we do actually is we make the patient an animal which is not sick. We are making a cancer drug, so we induce cancer in the mouse. We are working on tuberculosis, so we make mouse and infect a mouse with tuberculosis. So we are treating this. I mean, a lot of this experiment is happening through a painful treatment to the animals. Animal disease models also have low success rate. This is another aspect of the irony. Although we are doing so much of the animal experiments, but we are not improving. We are not really making good use of these animal sacrifices as well. So I used to work in Japan. They used to have, after every year, they used to have a ceremony to kind of pay some respects to the animals which have been killed during last 1 year. So that generates an awareness among scientists that we are taking life, and they used to pray for those animals. So these are some of the issues that the scientific world, the drug discovery process, the healthcare professions, the cosmetics research, they are facing. And if you look at this problem is even more compounded. This is not only that we are doing a lot of research, which is not producing the results that we want. It is a wasteful expenditure, but the environmental and ethical disasters are made worse by wasteful and irreproducible research. I mean, in 2015, there was a research paper that suggested that 50% of the research that we publish in literature, 50% of this, nobody can repeat them. So that means a lot of this resource wastage, that a lot of these consumables that we are sending to the environment, and a lot of animals and that we are killing every day are just for nothing. 50% of them, and nobody can even repeat those experiments. So if we look at what are the categories of the irreproducibilities, so you will see at the bottom, there is about 10% of them have a faulty lab protocol. And 25% is caused due to the data analysis errors and reporting. So I report that X does Y, this drug does this thing, this chemical does this thing, this molecule interacts with this, but nobody can reproduce that. So this is a complete waste, and study designs are faulty, biological reagents and reference materials are not correct. So nobody is able to reproduce those research. So this is a big, big problem, that not only that we are trying to find the haystack in a big husk, which is about 5%. But even that 5%, even this 95% or more, what we are doing is nobody even can reproduce that husk. Forget about the needle. So how is scientific research trying to help in these issues of having less wastage and having addressing the issue of animal models? So first, they are trying to provide alternatives to animal models, that you don't have to every time bring a mouse and do the experiment on the mice. So there are many alternatives. And one of the big alternatives that has emerged, and that is very promising, which has not reached its potential, is the in silico experimentation. Don't every time go to the lab and perform the experiments on the living systems, or even test tubes and chemicals. You can do those experiments on the data that you already have, and by tweaking the data, by doing a mathematical modeling, by doing a computational modeling or informatics based prediction, you can replace your in vitro, in vivo, and animal model experiments. So these science is trying to provide alternatives to animal models. And now a lot of people are doing it individually. This information may not be accessible to people. So another job of the bioinformaticians is to provide the databases and resources. So if I'm looking for a answer to a question, biological question, so that answer may already be available in some form or the other in the public domain through some other experiments, with the experiment which was done for a different purpose. So there lies a need for bringing that information together in the form of databases and tools in a centralized manner, in a standardized manner, putting in the right quantities so that the information is not repeated. So this is another way of the saving environment and saving ethical questions by providing information that, okay, you don't have to do this experiment. This data is already available. Or this experiment has not been done directly, but that another experiment has been done, which also provides answer to your question. So that is the focus of the bioinformatics databases. Biocomputing and bioinformatics are trying to help in creating these databases, providing this information that can be searched in a dynamic manner, sometimes search through standard language, sometimes search through the natural language. And that is where the language model-based search of the databases also becomes important. And the third thing is that to avoid the wastage, the predictions, the models that we develop on animals are, as I said, only 5% of them actually work on humans. So the bridging this gap, that the outcomes that come from the animals become more compatible with the outcomes that are expected from humans. So that is a lot of modeling and computing and bioinformatics is required to bridge the gap between the experimental outcomes that is obtained from the animals, the outcomes that are obtained from the test tube experiments and cellular experiments, in vitro, in vivo, and animal models on the one hand, and the human real systems on the other. So how can we bridge this gap so that the experiments don't have to be repeated and the fewer experiments have to be done? So these are kind of, broadly speaking, three areas in which the scientific research is focusing to also reduce the cost. This will also cost not only the cost to life or ethics, but also the cost to resources. And when you think about this, for example, this is a mind map for when somebody tries to collect the literature on the alternatives to animal models or alternative approaches which can to experiments. Then you think of, let us say, cosmetics is the industry point. So you think about what are the endpoints, what are the cost effectiveness issues of the system that we are studying, what are the regulatory guidelines, and whether it is banned in our country or not, what is the guideline in our country, in the country that we are conducting this research, and how it will collaborate. And the how now there may be some things which are not regulated, which may not have a legal issue, but they still may not be liked, or maybe may have questions being raised in terms of ethics, in terms of the extrapolation, the reproducibility, as I mentioned, and the variability that we are how much diversity that I want in my data. If I have results from 10 animals, will it also be the same result on another? So 10 models. So how many animals are to be done? So all these things these are the concerns. And then you also have the issues of the endpoints. What do we want to achieve? What are the endpoints? Toxicity studies, and out of them, what are the repeated dose toxicities? What is the dose issue? How much dose should be recommended, etc., etc. And then in terms of the experimental outcomes, these yellow bullets show that we can do them in vivo, in vitro, ex vivo, in chemico, and finally in silico. So how can we do in silico research to replace these other three blue spheres, yellow spheres that are there on the upper side? So now there are in silico methods. I've discovered something in the computers. Will I suppose I have a drug, and or if I have a cosmetic product, and I want to establish whether this is safe or not. So the safety of this, you have to test a lot of you have to do a lot of toxicity experiments. First, you have to test it on animals. You have to test it in on different cell lines, and then ultimately test it on human in terms of the clinical trials, etc. So before you bring this product to the market, it has to pass certain the regulatory guidelines. So these guidelines, whatever is, must be accepted. So I may do an in silico analysis and I may give a very good prediction, but will the regulatory authorities accept my product without animal test or without human clinical trials? So now this is the list of the alternatives to the cosmetics research. That is, this is one particular issue that out of the approved alternatives, 39 are in vitro, which have to be done in the laboratory. 6 of them have to be done in vivo, and some can be done in the chemical in chemico, and then just one of the technique is approved for in silico. So out of these, you look at what are the kind of toxicological endpoints that people are trying to look for. So they're looking for the eye damage. Will this particular cosmetic cause serious eye damage or eye irritation? Will it cause skin sensitization, skin corrosion, reproductive toxicology, skin irritation? Again, carcinogenicity. In the short term, it may look safe, but in the long term, it may lead to some carcinogenicity. So acute toxicity, basal cytotoxicity, phototoxicity, and genotoxicity, etc., etc. So mutagenicity. So these are the issues that you see. It somehow shows you also the inevitability of experiments with the animals and inevitability of experiments on humans as we do in clinical trials. So green toxicology is a subject that has emerged out of it when people are trying to develop environment-friendly methods for testing the toxicity of the compounds. These compounds may be cosmetic, and these may be the drugs or the medicines. And the principles are, there's a large number of principles on the slide. I don't need to go through each one of them, but the broad principles can be seen, a few of them here is it is better to prevent waste than to treat or clean up waste. So what is the idea is that before you start your experiment, so you choose an experiment in such a way that you do not produce too much of waste in that. So prevent wastage, and then use synthetic methods, and then use auxiliary substances rather than the real substances and the energy requirements, the raw material requirements, and reduce the derivatives that many a times when we do an experiment, some derivatives will be produced which will be just thrown into the environment. So how to reduce those, and the catalytic reagents, chemical products, all these issues. So these are those interested can look at some of these papers that I'm citing through. So a lot of funding is now going in this direction for computational toxicology, because computing experiments are really environmentally much safer than any of these methods. So if we can do something on computers, we don't have to do in the wet lab. And wet lab definitely will produce more toxicity, more waste products, and will also cause more ethical issues. The computer experiments will not suffer from that. So how can we move these issues onto the computers and developing the field of computational toxicology? But remember that computing experiments also themselves are often based on the data sets. I will come to this issue later, that how the data sets become important. So ultimately, what we are trying to do is not replace experiments. What we are trying to do is reduce the experiments. So trying to extrapolate the outcomes from my previous experiments to the new experiments. So I'm trying to reduce the number of experiments that are required to do a task. So for this, what we are doing in computational toxicology is we are trying to collect the adverse outcome pathways. That is the pathways, what are the genes and what are the cascading reactions that happen in human beings when some adverse reaction happens. What are the integrated chemical environments to study them, the integrated approaches for testing and assessment, because ultimately no drug can be approved without being tested. So these are some of the big projects that have been funded by the National Institute of Health in US, and similar projects are also everywhere in other countries. So there's one technique called the read across technique. So the read across technique is saving environment in some way, saving wastage in some way, because if we have a few products, then we should not have to do experiment on every product. And sometimes the information about that product, so the product here is a small molecule. So we try to fill the gap without doing the experiment by making a prediction, by trying to understand what is happening in this chemical list of chemicals. So what it does is it is a method of filling the data gaps for a novel, previously uncharacterized compound by relying on information which has been obtained from other chemicals and then putting them in categories of structures. And the underlying assumption here is that the similar structures have similar bioactivities. So we can just look at their structures. The structures are easily available without any consumable requirements. We can, even for small molecules, we can predict structures completely without ever having to characterize structures. We can compute structures, and the three-dimensional structures of small molecules can be very accurately estimated today. So if we can estimate the structures, and if we can find a relationship between the structure and the biological bioactivity that will happen as a result, then it is much easier to do an in silico based discovery of the new compounds or the new products. Or if we have a compound, let us say, and we feel that this is not soluble and we want to make this soluble, so we will, from the chemical analysis, we will be able to make it more soluble by tweaking with the side chains, tweaking with one atom or the other, and synthesizing those molecules in the laboratory with minimum cost. So this is what people are trying to do, this technique in silico read across. And how it works is like this, that it will take a graph-based representation of your molecules. It will take what is called the SMILES. SMILES is also a kind of representation of the compounds. And then you also take the images, you take the create a fingerprints from them, and you create the descriptors, their molecular properties, which are already measured. And then you use an AI or machine learning method and try to predict these properties, which are not known for the new compounds, which have not been studied. So basically what it does is for a set of compounds, I have the information, then I want to have information about many more compounds of that kind or slightly different from this. So I create a model from the known mappings, and this model will be later on used for directly predicting what will happen if I tweak this particular atom, if I change this so side chain of the protein, the small molecules, etc., and then we can accurately estimate them. So that is how AI and the bioinformatics are helping in this field, trying to improve these predictions, and the outcomes should match exactly what they were for the real wet lab experiments. So if I was trying to start initially, I was trying to start with 1 million compounds, and any of these 1 million compounds may be the compound of my interest. Through the in silico analysis, I can reduce these 1 million compounds to just a few hundred compounds that may be of my interest. So this kind of progress has already been made, and then these results we are trying to make more and more accurate. So the number of experiments that have to be performed, number of the potential drugs that have to be injected to the animals or in the clinical trials becomes much less. And the turnaround time, the time in which we can produce a drug, like you have seen a success story during the vaccination for the COVID, the time that it takes to discover a vaccine today is much less than what it used to happen in the past. It used to take years to develop a vaccine. Now we can develop vaccine in a matter of months. And perhaps in the future, we can produce vaccines on demand. You give the properties of the virus or a pathogen, you give the properties, and there comes the medicine, the potential vaccine. There will also be personalized medicine where you give the detailed genomic profile of an individual, and it will give you the best drug for this person. So this is how we are optimizing. The computers are saving the entire cycle of experimentation and incorporating the personalized, even the individual to whom the treatment is to be given. So going further on the topic of that I was talking about, the adverse outcome pathways. So in the field of bioinformatics, we are compiling the pathways that if we expose a particular molecular initiating event, which may be a compound or which may be any stimulus, it may be environmental conditions, maybe temperature or thermodynamic state. So if we have a molecular initiating event, and then there are key events that will happen as a result, then in this network, we can, through these networks, through the databases that have already been collected, they can map the properties of the molecule to the adverse outcomes that are likely to happen from them. So now, as I said, all this comes from the development of powerful data sets, because information, as I said, millions of experiments are being done routinely every year in the entire world, and so many scientists are are producing this data. So there is a need for people to sit together and collect the data. Sometimes even the data has the problem that names of the compounds or the names of the plants or the animal models or the diseases or the symptoms that people are collecting, even their naming is not standardized. So the first big challenge for the field of bioinformatics is also to develop high quality literature and other databases. And there is the term that has come, that is data warehousing, where the multiple people have developed their own data in what they have collected in their laboratory or in their company, and what others have done in one country, in one state, or in one organization. People have developed their own data sets, but the data sets taken from one source may not be comparable with another. They may have been named differently. In some places, they may have mentioned the temperature of the person. Some they may have said just fever. Some may have said high fever. Somebody may have used a different range for high fever. So these the different types of biological or biomedical data that are collected with different notations, different details, and putting all that information together in a standardized database as a warehouse where people can ask questions of that they want to ask is important from just the information which is already available. So what we do today is, if we can't find that information, we do an experiment. So that is how, by proper databases, we reduce the number of experiments. But databases are not just important for the referencing of information. Databases are also important for the modeling. no AI or machine learning based methods work on databases. They say your models are only as good as your databases are. So we need this data to, if we cannot generalize, we cannot do machine learning. So for machine learning, we need high quality data sets, and that they have to be properly quantified. They have to be properly labeled, as we say. So therefore, machine learning and AI requires high quality databases. These data sets are the starting point for any ML based or deep learning based approach. Then data sets are also there for meta-analysis. That is, multiple experiments have been done to ask the same question. As I say, many of the experiments are not even reproducible. So if 100 people have done, for example, 100 people want to know what are the genes which are overexpressed in influenza, so that a drug can be designed for that particular gene. Now, if independent labs have produced different outcomes, and the genes that one group has identified do not match with the genes that another group has identified, this is a big challenge what to do actually in the form of drug discovery. So this meta-analysis of the data, how to compile the evidence, independently collected evidence, and how to remove the noisy data, how to remove this reproducible data from the reproducible data, how to make this quality of the data, and by integrating multiple experiments, what is called meta-analysis of the data. So to develop a consistent and holistic perspective from the information that is produced independently. And as asking novel questions, somebody may have never done this experiment. Okay, what happens if this gene is removed? What happens if an individual has this particular mutation? No individual may have that mutation so far, but we can also already predict. We can ask novel questions, like in the case of personalized medicine, we know that this individual has this genetic profile, and this individual has certain mutations in some genetic locations. So will the same drug work for him, or will this person be drug resistant to that? So these kind of questions that we are asking, no experiments may have been done on those questions to answer those questions, but if we have good data sets and if we have good models, we can extrapolate and we can try to find answers without going to the laboratory. So now in this effort, this just to give you an idea about the amount of effort people are doing, this is the number of data sets, big data sets that people have developed for chemistry data, 80 data sets, which are relevant for these animal alternative methods. These are all the data sets that people have developed to work on alternatives to animals. So data sets of protein-protein interactions, omics data, pathways data, patenting data, so that even if I know, so I have something knowledge, but this particular compound is patented, so I cannot really move on my research. So then environmental factors and the nanomaterial toxicity, et cetera. So this is just a list of data which I have also provided the reference. So now there is this in US, this is a national toxicology program, and they have developed this whole big project for alternatives to animal testing. And you can search what are the papers that have been published. So this is one such data set, which is database, which is very important. And people trying to do lab experiments can first go here. Lab experiments on animals can first go here and try to find, is there an alternative to what they're doing here? So these are a list of data sets. I'll cross them. So the issue, another issue is when we are trying to use the innovation, or as the theme of this conference is innovation or AI. If we are trying to use AI for these questions, then we use data curation. And again, we have to bridge the gap between what we are predicting and what actually happens in the lab. Unless this gap is little or reduced, we will not be, nobody will trust our computational outcomes, and a lot of this will just go waste. And we will continue to do experiments in the laboratories. So alternatives to these laboratory experiments require powerful, cautious application of machine learning. And most important of them is to avoid overfitting, trying to give very accurate results for the data that we have, and the same models do not work at all on the new data. So this problem of the overfitting is one of the biggest problems when we try to use machine learning based models for any replacement of lab activity. So there are efforts. I give you some examples. For example, there is an effort going on in terms of the prediction of human phenotypes from animal models, and people are constantly trying to make predictions, and machine learning AI is helping. So for example, this was one competition that was held by this IBM and other groups. a dream challenge, and this dream challenge gave the data of human phosphorylation and gene expression from rat data. Sorry for the typo. So these two data, and then they had hidden the data from the human, some samples, and they provided some data for the rat and human, like the red bars in this slide that you see. This data was hidden, and this scientific community was given this data, and they were asked to predict this data P part from the rat to rat, from some experiments on rat to other experiments on rat. Can you make a prediction? And can you make a prediction from experiments done on rat to the data collected from the human? So this kind of competitions are going on, and a lot of AI and machine learning effort is going in the direction of improving these connectivities. Then this is another very exciting area of research for the bioinformaticians, and which has again holds a great promise of replacing the animal models and other experimentations, which is called the gene knockout. So those of you who don't come from biology background, gene knockout is basically you have an organism, you have an individual, and it has an X number of genes, and we want to see where, yeah, is there something? So we want to predict what happens if this gene is removed. The purpose is that we want to know the function and the role of this gene in causing a particular biological outcome or medical condition. So typically, for this is a usual experiment, then people develop the genetically modified or knockout mice, as they call it. So in the knockout mice, they create a next generation of the mice by removing a gene during the replication process. And that is how they see that what will happen when this gene is removed. Now, this is a different matter, that it may do something for this mouse. It may not do the same thing for the human. But nonetheless, this is one of the largest number of experiments that are available, that are carried out on animals. So now, since so much of experiments have been done, and so much experimentation on not only knockout, but other gene expression analysis have been done, we have data when this gene is expressed, what is happening to other genes, etc., etc. So people are trying to compile data from multiple sources and trying to develop methods so that scientists can carry out an in silico knockout. That is, within the networks of the genes that we have in our data, if we remove these genes, what is the predicted outcome? Purely computationally, we can predict. We are not really that advanced yet to be able to accurately predict the outcomes, but the number of experiments required to verify has reduced. We are able to now use in silico knockout and do fewer tests afterwards so that the outcomes can be tested. So this is the kind of research people are doing, and these are the directions in which we are trying. And another area of AI that is helping a lot, and that again holds a great promise in terms of the environment, in terms of the safety, in terms of the cost, is that of super resolution. We usually do, for example, if somebody is trying to do an MRI, trying to understand a brain disorder, then the technique is to take the medical resonance imaging and exposing the individual to very high intensity of radiations. So now the research is that you should not expose the individual to such a high degree of resolution. We can collect the information at low resolution. So we take low resolution images and then use the deep learning, because the images, the pixels or the locations of the brain do not act independently. There are some relationships, and if I have this information, I can study these relationships and I can computationally generate a high resolution image. So converting a low resolution data into a high resolution data reduces the burden on the data collection, that we do not have to collect our data at such a high resolution in such greater details, collect the data in less detail, collect data at low resolutions, and we can regenerate what is actually there. So this is another fascinating area which is being applied to understand the biological diagnostics and treatments and finding the best therapeutic approaches. So now I present some of the work that we are doing in our lab. So we have mostly spent, I have spent most of my time. I'll take some of the slides that I have here. So in our lab, we have developed some of the resources. As I mentioned, we need high quality integration between data sets and experiments. So this is one such approach that we did, an exciting research that we published in Nucleic Acid Research, which was an integration of gene expression and sequence. So people have been, if we collect some information on the genomic sequences, or if we collect some information on the gene expression, and the protein level. So typically these experiments are done independently, and after the data has been collected, people try to create a picture what is happening from the MRI images, from the mutation analysis, from the genomics, from the other sources. So one thing is because everybody, once we have a genome, then this genome has information about all the genes, but and we know that this gene performs this function. But it is actually in the cellular condition, the gene sometimes does this function, sometimes does not do that function. So there is a difference between the potentiality of a function and the actuality and actual happening of the function. So in order to understand what actually happens, this is collected in the form of gene expression. What is actually possible is collected in terms of the sequence, function annotation, and the structure of these proteins and genes. So integrating them, we have collected this data of DNA binding protein, one particular very important type of group of proteins which play all the role in gene expression. So in integrating the information that comes from the gene expression profiles and from the sequence and structure analysis, so we have created this database, and we try to show that if we do, we can actually predict function directly from sequence without actually going through the structure. So integrated analysis can produce, again, cut down the route to discovery, path to discovery can be shorter. So I'll not touch upon the technical details of this. I'll come to another issue that we are dealing in our lab, that is trying to predict the function of the protein. So if I only know because most of the biological functions in the human body or in the living systems are carried out by proteins. And these proteins are encoded by these genes, and the function of these genes is still unknown. For a large number of genes, the function is still unknown. So how can we annotate them, or how can I assign them functions from the existing information without having to do all the experiments? So these experimentations can be again replaced by AI-based models like the CNN. So one particular problem that we are solving is to predict the DNA binding proteins, and we are able to predict them with a high degree of accuracy when we use this approach of the CNN, which is again a deep learning based model. So this is the work that we have published recently, and this is and just I'm giving you the list of the projects that we are doing to give you an idea how the experimentation is being replaced by the computational experiments and how we are contributing to this. So this is another field of experiences. So this is, as I said, the feature selection. So if I have, say, the particular image or a gene expression profile coming from one location of the tissue, say, let us say I'm collecting the data from different locations of the brain. And when I collect the data from one particular cellular locations of the brain, and when I collect the data from another cellular location of the brain, and if I do not know from where this data is coming, can I put them together by a computational method? So when I try to put them together through a computational method, do I need to have the expression data for all the genes, for all the points? Do I need such a detailed information? Or can I just select a few genes which are critical for my for the location? So this problem of finding a subset of genes which can provide me the information about rest of the genes. So this is an area of active research that we are doing, feature selection, and trying to reproduce the entire gene expression profile from a subset of genes. This is an area in which we are contributing, and we have some exciting results on this. So we participated in the DREAM competition, and in this DREAM competition that I was just mentioning, our team stood among the top 10 for a large number of questions, the sub-challenges, as they call them. So one of these sub-challenges, so GEEKS is the name of the team that our students went with, and we were in top 10 teams where the top world best scientists were participating in these competitions. So then finally, again, the drug discovery process, where we are trying, interest is in silico prediction of toxicity, what will happen when this drug is treated. One problem in the drug discovery process is that discovery happens when they say, okay, this is the target protein or target gene, and I want to make a drug based on that. So this can be tested in the test tube or in silico, but usually this gene is tested in isolation from the real living system in the cellular condition, as you call it. So in the cellular condition, its behavior may be totally different from what its behavior was in the test tube. So find that, and that behavior changes because of the toxic interactions, because of the, so now people are trying to develop methods to predict that this compound is likely to cause such and such impacts. So a relationship, a connectivity, as I call them, the connectivity between the small molecules or perturbations, gene perturbations on the one hand, and the entire system, the not one gene, two genes, but the entire system impact caused by the drug, and create a big database a priori so that everybody who wants to test whether this drug will be good for me or not, they can pick up the drug from this library and then try to work on the next level, rather than trying to go through the entire process of toxicity prediction and other things. So this project is a big project, again, led by the Broad Institute and others. And the CMAP project, in this project, we are contributing by developing existing databases that already exist in the gene expression omnibus. So we collected the data from, so millions of the samples have been deposited there. So we have integrated data from these many number of samples. As you would see, there is one gene expression platform called the Epimetrics GPL 570, which contains experiments from 135,000 experiments. And the data from another source is 15,000 experiments, 40,000 experiments. So these are all the row shows how many experimental data, how many experimental profiles of the gene expression are already available. So we need to integrate them. We need to complete them. Some profiles are collected on these 1,000 genes. Some are collected on another 900 genes. So how we put this together, and so that every gene information can be collected, we use the AI and deep learning methods to predict such relationships. So I think I've run out of my time, is it? The chair? Do I have time? So I'll just take 2 more minutes. I can't hear you. I think your voice is echoing, but I'll just take 2 more minutes. Okay, so this is another project that we are doing, is to automatically predict the erythemia from ECG signals using digital technologies, so that we do not have to go to the doctor every time. So you see, the environmentals, we are saving environment even when we are not going to the doctor. By just consulting our doctor on the internet, we do not see how much environment we are saving. We are saving our transport, we are saving the petrol, we are not driving. So physical movement also is a burden on the environment. So some of these burdens can be reduced by automatic diagnostics. Online consultation is one, but using a software is another. So this is not just the question that about environment. This is a question about the availability of doctors, experts, and how quickly and how accurately we can predict the erythemia from ECG signals. So this is some of the software that we have developed. So I'm kind of putting this all in a summary, that rapid advances in medical and biological sciences have helped us improve the healthcare longevity and agricultural productivity. With the help of AI, the discovery process is now very promising, and it is likely to take bigger leaps. The life expectancy that I was showing was a kind of major breakthrough came in science during that time. Now AI is a major breakthrough, and we expect that the next part of the graph will be even sharper. So animal models, consumables, and other resources spent on research can be drastically reduced by better computational modeling, extrapolation of knowledge from current data sets, and better performances. So the concluding thought is, on the right, I have this graph that is the projection of what will be our lifespan in, say, 2100. This data I got only from Japan. They have done this. I couldn't get data from India, but these are the projections that they expect that people will live on the average 100 years in 2100. And this 2050 is already there. Life expectancy there is more than 80. We still have to do catching up with them. Japanese are one of the most longest living people. So the concluding thoughts on this is that we are expected to live longer than any human generations have ever lived in the past, except for the mythological or other belief systems. But as far as the scientific detailed information on the life expectancy is concerned, last several thousand years. So as I said, 1950, the life expectancy was just 60 years, even in Japan. So, and now we are more civilized. I believe that we are more civilized today. Our civilization has understood, is living much more peacefully than people used to live in the earlier times. And we are better informed. We have more knowledge than any of our ancestors. And simultaneously, our consciousness is growing, and perhaps it will continue to grow. We will be more harmonious with one another and overall life on the planet. This is my hope. And thank you. With this, I conclude. Thank you. I stop here.
Now I would like to invite Professor Jyoti Misra ma'am to propose a vote of thanks to Professor Shandar Ahmed sir. Jyoti ma'am. I think she is busy. Okay, Professor Ahmed sir, very excellent talk.
Thank you.
And you spare a valuable time and giving a very informative talk on artificial intelligence in bioinformatics, and is a very informative for all the students. Now I invite the students to take any questions from our expert Ahmed sir. Anybody have any question? If you have any doubt, any question in your mind regarding any research, how to collect data, anything else, whatever you want to ask, the session is open for question answer. Okay, sir. Thank you very much.
Okay.
for
Thank
your,
you.
You spare your valuable time.
Thank you.
And very thankful to you, sir.
Thank you. I'll take leave.
Next time we want to meet you, sir, face to face.
Yes. Okay. Yeah, I understand. I think part of the reason that there are no questions is because of this lack of physical sense of
yes,
being
sir.
together here. Okay. Thank you. I'll take leave then now. Thank you.
Yes, sir. Thank you, sir. Okay, now we are going to start the next session, paper presentation session. In these sessions, we have two experts, Dr. Pankaj Dadit, sir, and Professor Sandeep Saxena, sir.
Thank you, sir.
Welcome, sir, in this session. Sandeep Saxena, sir, welcome.
Thank you, sir. Thank you.
Professor Sandeep Saxena, sir, presently working as an HOD and HOD in Computer Science and Engineering Department and professor in GNIOT Greater Noida. Before this, he worked as a director IQAC, professor IT at IMS Unison University, Dehradun. He received a PhD degree in CSE from NIT Durgapur, West Bengal. He has more than 16-year teaching and research experience. His area of interest and research includes security and privacy in blockchain technology and cloud computing, architecture design for cloud computing, and access control technique in cloud computing. He has performed the role of a key member in more than 20 international conferences as a keynote speaker, organizing secretary, organizing chair, session chair. He has written 4 technical books and published multiple research papers in reputed international journals and conferences. He has published more than 35 papers in reputed peer-reviewed journals, conference indexed by Scopus SCI with high impact factor. More than 10 patents published and 3 patents are granted. He is participating in multiple professional societies like IEEE, IAASSC, senior members, lifetime member in CSI, lifetime member in CRSI. He has been working on various research and development activity, university syllabus design, and mentoring innovations and incubation related developments to students. He has served as a reviewer and member of editorial board of several prestigious conferences, journal transactions like IEEE, Springer, and other Scopus Index international journal. He was contributory to various prestigious accreditations body like NAC, NBA, and others. Again, once again, sir, we welcome you in this session, Professor Sandeep Saxena, sir.
Thank you, sir. Thank you very much, sir.
Now, once again, I welcome Dr. Pankaj Dadit, sir. He is currently working as an associate professor and deputy head in the Department of Computer Science and Engineering, SKIT College, Jaipur. And he has published more than 18. He has more than 18-year experience in teaching. He has published 23 patents and granted 6 patents. Okay, he has published and granted 5 Australian patent, 1 German patent, 1 South African patent, and 1 USA patent. He has also registered and granted 2 research copyright at Registrar of Copyrights, Copyright Office, Department of Promotion of Industry and Internal Trade, Ministry of Commerce and Industry. He has presented 62 papers in various national and international conferences. He has 72 publications in various international and national journals. He has published 9 books, 32 book chapters so far. He is a member of many professional organizations like ACM, CSI, International Association of Engineering, and ISTE. He has been appointed as a PhD research supervisor in Department of Computer Science and Engineering at SKIT College, Jaipur. He has also guided various MTech research scholars. He has chaired technical sessions in various international conferences and contributed as a resource person in various faculty development program, workshop, STTP, and conferences. He is also acting as a guest editor of the various reputed journal publishing house, conference proceeding, Bentham ambassador of Bentham Science publisher. His area of interest include high performance computing, cloud computing, information security, big data analytics, artificial intelligence, intellectual property, right, and Internet of Things. Once again, Dr. Pankaj sir, we welcome you in these sessions.
Welcome, Pankaj sir.
Thank you,
Welcome,
sir.
Sandeep sir. We are here to enjoy this session, and I know
Thank-
that you're going to add value to this session.
Thank you, sir. Thank you, Dr. Chitresh Banerjee, sir, Dr. Sandeep Saxena, sir, Dr. Yudhvir Singh, sir. Thanks a lot, sir.
Now invite Pratibha Sudhakaran to present your paper.
Yes, sir. Can I share my PowerPoint?
Yeah, you can share your screen. Pratibha, you have 10 to 12 minutes to present your paper
Okay, sir.
and conclude your all importance, aim, objective, and findings, methodology, each and everything within this time.
Okay, sir.
Okay.
Yeah.
Now over to you.
Thank you, sir.
Okay.
A very good afternoon to respected dignitaries and the entire panel at Amity University, Rajasthan. I would like to present my paper, which is entitled Recurrent Neural Network Approach for End-to-End Deep Learning of a Continuous Speech Model. My paper ID is AIAIA 2314. So the process flow is I would like to introduce my topic, that is speech recognition, giving you an overview of RNN CTC language model, the experiments that have been conducted with regard to the database. The database is in Indian English, and the metrics and the accuracy of the system. So what is a speech recognition system, which is also known as the automatic speech recognition, is a technology which is quite complex and challenging task for us researchers. It's a technology that converts the spoken language into written text. So while humans can effortlessly understand and interpret spoken language, teaching machines to do the same is a significant technological feat, I must say. There are several reasons why speech recognition poses challenges. That is variability in speech, the age, the gender, the background noise, the vocabulary, the language model, the constraints in each language, the contextual understanding of the language, whether the person is dependent or independent speaker models, the data availability, and the quality. But despite these, due to the recent advancements in machine learning, deep learning, and neural network, we have been able to accomplish to a large extent progress in this particular area. So if I just give you a small background check, we were initially dealing with small vocabulary. We have gone up to medium vocabulary today, larger and very large vocabulary. So we have moved from simple isolated words to connected digits and words, to connected words and continuous speech. And now we are also getting into spontaneous speech. So this paper is basically what advancement we have done from the traditional ASR techniques to an end-to-end ASR technique. So in the traditional ASR technique, when we were using open source tools, like the one that we have used was Kaldi, there was a huge process, that is we had feature extraction process. Then we had acoustic modeling, we had pronunciation modeling, wherein we had to create an entire dictionary based on the phonemes and the words, and the language modeling, which also included the decoding, which was a huge process, time consuming process. Now when we stepped into the deep learning techniques, we have seen an incremental, kind of a lot of reduce in the complexity of our architecture, as well as it has improved our word error rate and a character error rate, and therefore has improved the performance. So here we are introducing to you to end-to-end ASR system, wherein we have used RNN for this technique along with LSTM and as well as CTC. So if we look at the previous speech recognition system, as I said, we used to have a feature extraction. We had pre-processing of the speech signal. Then we had feature extraction where we were using MFCC or LPC or LPCC. And then we were going for the acoustic modeling. Then we further went into language modeling, decoder, and post-processing. So right now we are using a neural network-based system. This is the Bedou's model of the RNN model, where we have 3 fixed layers. Then we have hidden layer. This is a bidirectional model. The one which we have incorporated is a unidirectional model. So an end-to-end deep learning in continuous speech recognition basically aims to learn the entire mapping from the audio waveforms to the transcribed text in a single integrated model. So it is RNN is having many architectures. It can use the LTC, that is the connections temporal architecture, or it can also variably use the LAS, that is the listen, attend, and spell. And here it also uses nowadays the CTA, the connection temporal attention architecture, or the transformer based models. So we are looking at RNN here, which is a class of neural networks that are particularly well suitable for processing of the sequential data, that is including the speech data. So here what we do is we take each utterance is a time series. So we convert it into a vector. So y of t I have taken as p of the probability of occurrence of that in your entire speech model. So in this particular model, we have our entire the different sequences or the different so here h of t is applicable to the first 3 layers, and then we have h of f, that is for the forward propagation, and h of b is for the backward propagation. Here our G stands for nothing but the linear activation function, and W is nothing but the weight matrix of each layer, and the B stands for nothing but the bias parameter. So this is for the forward one, this is for the backward one, and the last one. So here we have taken 3 fixed layers, and then the hidden layers are above. So this is how the conventional RNN systems will look like compared to our traditional system. So RNN basically here uses the back propagation through time to back propagate through the network to adjust its weights so that we can reduce the error in the network. So this is basically like a prediction model. So to measure the error in the prediction of this model, we compute the CTC loss after we have computed the predictions. So if I look at a typical RNN system, or we use what is called as the BPTT. So in the BPTT, that is nothing but the back propagation that we have used in this particular model. We are taking different the back propagation through time. So when we are taking, say, for example, in the case of E3, so this RNN uses a technique which is called BPTT, that is back propagation through time. So through the network, when we are doing this back propagation, it helps us to adjust their weights so that we can reduce the error in the network. So here, for example, if I take, say for the example of E3. So when I'm taking E3, so if we consider the derivative of this E3 with respect to S3, then we can say that this X3 is also related to S3. And we can also say that this S2 is also related to S3. So we have continuously taken the partial derivative throughout the weights. That is what these equations are here. So similarly, this S3 is related to S2, similarly relates to S1, similarly related to S0. So our final equation, we can put it as in this format. So what is this? This is acting like a chain rule. And we are accumulating all the dependencies with their derivatives. And then we are using it for the error calculation. So as we understand how this BPTT works, this is basically all about how RNN adjusts its weights and reduces the error. But what if we have a huge network? Say, for example, we have layers which are hundreds of layers. Then we end up calculating the partial derivative of such a huge multiplication. And at that time, the error that we face is called as a vanishing gradient problem. So RNN, to predict an output, will definitely use a sigmoid activation function. And this sigmoid activation function will become very, very small. And hence, it is difficult for us to predict the output correctly. Hence, one of the techniques that we have used in our flowchart is that we have included the RALU, which is called as the rectified linear unit. So the RALU, it is one of the ways to kind of remove the vanishing gradient problem from the RNN. So it is either going to initialize the weight matrix properly. So this is an activation function that introduces the property of non-linearity to a deep learning model and solves the vanishing gradient issue which we were facing in RNN. So basically, we moved from a traditional ASR network, we incorporated RNN, and when we were facing an issue with RNN, we incorporated what is RELU, and then we were moving towards LSTM. So now RNN also had a problem like a long-term dependency of words. I would like to give you an example. Suppose the word was the clouds are in the, so the system, depending on the context that is cloud, understanding that the context is cloud, the next word prediction used to be, the next word would be sky. That means it was taking it properly. It was taking the output as the clouds are in the sky. So our RNN model was basically able to predict the sky here. But if the sentence was bigger, like Jane was born in Karnataka, Jane used to play for a women's football. She has topped at the state level examination, but Jane is very fluent in dash. Now, ideally, it should be fluent in Kannada. But this is a problem with long-term dependency of words. It may not remember that the context of Karnataka. It has forgotten that the context of Karnataka. So when the sentence is longer, we did face a problem, and hence we came to the LSTM. So LSTM was one way to solve this particular problem for us. So in the LSTM, the introduction, there are 3 gates. That is the input gate, the output gate, and the forget gate. So in which the forget gate basically takes care of what information needs to be dropped going through the network. In this way, we can have a short-term as well as a long-term memory. So what we have done is we have incorporated LSTM in RNN. So we can pass that information through the network and we can retrieve it even at a very later stage to identify the context of the prediction. So today when I say mouse is really good, the mouse is used to, so it will be for a computer, it is used to scroll for the easy use of a computer. That has been possible. Then we have incorporated the CTC algorithm into this. So the CTC algorithm here is allowing us to assign a probability for an, say for a, say I will say y is equal to CAT cat. I want y and the y. which is to align the output character to each character. The alignment was initially done something like CCAAAT. Now what we are doing is we are introducing, in case of the CTC algorithm, wherever there is a repetition of two likely, that is same letters, we have introduced what is called as a blank token. So the blank token is like an epsilon symbol. So if Y has, say, two of the same characters in a row, then a valid alignment that is such that a blank token has been introduced between them. Now, with this rule in place, we can actually differentiate between alignments, and which collapses to, as you can see, HELLO. Otherwise, what used to happen is we would just end up with HELO. So the CTC algorithm or the CTC alignments gives us a natural way to go from the probabilities at each time step. Because when we used to give just the occurrence of, say, CCAET, at every time step, it was getting an output. So it was the probabilities when we were giving at each and every, it was creating a problem. That's why we have used from literature the blank token concept used in CTC. So this is the entire expression for the CTC conditional probability. So from the traditional ASR system, we have incorporated RNN to, from the problems that we have incorporated in RNN, we have come to the LSTM and the CTC. So the experiments that were conducted, the data set. Yes, sir.
Hello.
Yes, sir.
Sorry for interpretation. You have more than 10 minutes. So kindly conclude your paper presentation in 1 minute.
Yes, sir. Yeah, I'm coming to the experiments, I guess. So in our paper, we have used a data set of Indian English with both male and female speakers with 45,000 tokens. And the model is a deep speech model, which has 5 dense layers, 1 unidirectional RNN layer, and total of 6 layers. This is the entire flowchart of the unidirectional RNN-based deep speech model. And we have compared the result also with our first, when we were using the traditional speech recognition system. So in the metrics, we always calculate word error rate, which is the sum of I'll directly come here. Word error rate is S plus D plus I divided by N, where S stands for substitution, I stands for insertion, and D stands for deletion. So let me give you an example. So in our result, we have got like, I bought this fork with me from here. So the output S is a substitution. So some places, instead of I, here the has been substituted. We have examples wherein there will be some sort of insertion also. Instead of Bohemian, it has taken bowl him. So depending on whatever I's, S, C's, and D's we have got, we have calculated the word error rate for both male speakers and female speakers. And as we know that RNN basically works, or it takes data sequentially, word by word, or letter by letter, or character by character. So it's basically using of memory elements in one neural network. So with that, we have got the following results in word error rate and in the character error rate. So the observations and conclusions, these are my original, and the output I have highlighted places where the errors have occurred. So from this, I would just like to conclude that we do face a problem when we are talking about homophones like here, here, R, or night, night. We also have seen good results in case of transcriptions like salutations or possibility or shuffle, but it does not basically transcribe proper nouns properly, like Tenali Laman, or a name when it happened, or it has to predict a particular name. So the conclusions to the paper are, we have introduced a recurrent neural network that can perform character level voice transcription with little pre-processing, and no explicit phonetic representation or dictionary is required here. We combine the network outputs with a language model during the decoding process to a unique objective function that enables the network to be directly optimized for word error rate, and the state-of-art accuracy for speaker independent identification for the Indian English corpus for continuous speech has been incorporated. And we have used all these references. And so that is from me. Thank you. Questions, please.
Hello.
Yes, sir.
So what is the main problem in this?
Sir, in the main problem, we were actually, we have used what is called as a Kaldi based model initially. It was, we had to create a dictionary initially, which had, it was very time consuming for us to have a dictionary, but, and it used to be based on the sound or the phoneme only. But when we shifted to RNN or a deep speech model, we used the probability occurrence. That means the next word depends on the previous word. So in this case, we were when we were training the model, it was the dictionary was kind of being created or trained on its own. While in case of the previous model, we had created 22,426 words dictionary, which had to be done manually. So that was giving, or we can also use a kit called SRLM kit, which we were using. But compared to that, the deep speech model is giving us a better result, sir, compared to the previous model. So that was a problem that we had faced. So the dictionary, the acoustic modeling was more tedious for us. But in this case, because of the probability of the previous words that we are using, and as we are building the database, the errors were minimizing, sir.
Okay, okay, thank you. Sir, any question?
No, sir.
Okay, thank you. Next.
Thank you, sir.
Okay, now I would like to invite the next presenter, Monica Anand. Monica, are you there?
Okay, call next one if it is not.
Okay,
She is not available.
Next is Kailash Bhosle. Kailash Bhosle, are you there? Okay, now I would like to invite Satish Kumar. Satish.
Satish, are you
Yeah,
there?
sir.
Yes.
Yes, anyone want to present? He or she can present.
Satish, please share your screen. First, open your PPT, share your screen. अभिषेक तो है ये तो सतीश है नहीं नहीं उनका कह रहा मैसेज तो मैंने पूछा उसको मोनिका आनंद और उसको फोन करो मोनिका और किसको
Okay, it is visible. Make it slide so more. Yes. Yes.
Satish, we cannot hear you. We can see your PPT, but we cannot hear you. Check your mic. And ask to Satish, am I audible? I will make a chat for you. Better option. Everyone. Just wait a minute. मैं करूँगा। अभिषेक को बुला दो।
Till then, I would like to invite next presenter. Kumar, are you there? Abhishek?
Hello. Hello. Hello.
Yeah, I'm audible.
You can start your presentation.
Actually, sir, I am unable to upload my PPT, so just give me 1 minute.
I think there is a technical issue in the.
So here I am sharing, if my screen is visible, sir, here I am sharing my PDF file. Is it visible?
PDF file is visible, but
Yeah.
not in presenter mode. It is very. Kindly zoom it.
It is not readable.
Wait a minute.
Readable. Kindly zoom it.
I'm doing that. Just a minute, sir. Now it's visible, sir?
Yes.
So hi, good afternoon, everyone. Sir, am I audible now?
You are audible.
Yeah. Hi, good afternoon, everyone. I am Abhishek from Department of Design, Manasthali Vidyapeeth, and I am going to present my paper on artificial intelligence, impact on creativity and design, ablazing org search. Basically, why my thought is why I write this paper is actually AI is a boom word right now. It's a buzz, and most of the time in creative field in design, everyone is talking about that AI will reduce the job, AI will do all these things, AI will do whatever it wants. But AI will do. My question is, do AI will survive or do AI will succeed? So here I have that written. Artificial intelligence become an exceptionally popular approach to problem solving, not only among the creative fields but across all the disciplines. But in design, actually, due to creativity and that all of the human thought, AI will never succeed in design. Most of the time, like if you are a very good prompter, you can create whatever you want from AI, but you can't go with the basic principles of AI. So in AI, if you have a good prompting skills, you can do whatever you want. But when we go for the design principles, it's not possible. Like just for example, a few days back, we have a morph image, a lot of morph images of that Geeta Fogart and all. Might be everyone has seen that in the news channel. It was on prime slot, but that image was morphed. So that is a huge misuse. But a designer can't do that. For that, you need a lot of skills. And if you are a good prompter, you can do that. So mine is, so the word, let's introduce the artificial intelligence in design. The word artificial does not require any introduction today. But in 21st century, everyone has some basics of their skills. They are doing all these things. So let me change that. This is my introduction, and each and every company, like giant companies of design industry, Google, Adobe, Mahindra, they all are Autodesk. They are all are betting each other for who is giving, who are going to give the biggest artificial intelligence platform to the designers. Directly or indirectly, it's just they are pushing us to use all these things and forget about the basics, the principles of design and all, and start doing with the software. Forget the manual, old school learning, and whatever you want, you can just do with the software. So all these companies are there who are doing the new researches, and just they are proving like the Apple that spectacle computing. Last month, Apple has launched that. So that was just by their neural. Means you can just control all these designs by your brain. What you are thinking, you can see on your the screen. Hello. Sir, is my screen is visible?
Yes, visible.
So what is the advantage of artificial intelligence in design? Design is not a new thing. It's for decades. Even if you see the earliness, they have design on the tapes and all. But in present scenario, in 21st century, or I can say that the generation which we call Gen Z generation, they are getting changed. They are not very handy with the basics of design, whatever the principles, why design is. They are very well aware with the software. They are very well with aware with the mechanism and all. So they are very well with the system, well versed with the system. They don't know what is the basics. Just if you say them, you have to do this, they will search on Google and they will come with the replica. That's why some advantages are there, but moreover, disadvantages are there. Here I have put two examples of Raja Ravi Verma. Raja Ravi Verma took I think 7 or 8 years to create the image of Almighty God Rama or that Maharana Pratap image. It took 7 to 8 years. He did a lot of studies, live studies, then he made a lot of people, and then he comes with that, okay, Maharana Pratap or Lord Ram, it looks like that. This is his portrait. But I think 7 or 8 months prior, there was one image of Lord Ram surfaced on the internet of at the age of 21 how Lord Rama looked. So the artist in his interview told me it took 2 to 3 hours only to create this image of Lord Rama when he was 21. So these are the basic differences between the software and the process of design. So the images of generation, they are rocking our industry. Like few days back, we have one movie called Brahmastra. In Brahmastra, they have used extensive use of artificial intelligence to create effects and all. But see, there is no logic of that because they don't have the design sense. That's why it's a super flop movie. And the intelligence, whatever the data we have, it's creating the design. So whatever might be it's a data, wrong data or right data, nobody knows. So whatever it's there, whatever the data is there, the design, whatever by prompt or by Google Bard, Chat GPT or neural design, whatever it is, it can be there. And one of the good example is that the natural intelligence Nikon camera. Nikon has launched one campaign which is called natural intelligence. Because in design, human intake is important, digital intake is also important, but after all, how much emotionally you are connected with your design, that is very important. So just last month, Nikon launched one natural intelligence design. He coined his own word as print and outdoor camping that goes against AI, presenting photos of natural places which in our world data is stranger and than fiction shot by photographers using Nikon cameras. So what Nikon has done, he has sponsored that plan. How just take this camera and visit the places where what places you want, and then click the images, and they have shown this. You see, this is the emotional intelligence, or this is the natural intelligence, which can't be done by the artificial intelligence. You just prompt, okay, I want this, but you can't get this. And one of the best example of that is that artist, that image of a vulture and a boy. So just some of the people, they want to recreate that, but it's highly impossible. They can't get the emotion. So in my case, AI is a bird, but AI can't replace human. AI can't replace the artist. Yet the benefits are there, but it can't replace it. So maybe in coming years, in few 5 years, we can see that in AI workflow, like in design, Adobe is the giant company. Adobe has launched lots of tools in that Animate, CC with artificial intelligence input. So the benefits are new design tools and workflows. We can find out all these things, like we don't have to go for the manual or old book theories and all. Whatever you are thinking, may be there on your screen, but the workforce will be there, but generative design can do whatever you want, but the emotional touch can't be there. Then the creativity will enhance, of course. Researchers are working on the way of AI to directly enhance human creativity, but the creativity will be enhanced else if you have the data. And then generative design concept and ideas, iterative design, generative personalized and customized designs, and documentation of design, new design jobs and roles. My focus was that because a lot of buzz are there, everyone is asking about do AI will take our job. So my thing is that AI will not going to take your job. Yeah, it will be just provoke the thoughts. It will just you have to be on your toes to perform your job. So new design in AI might be after 5 years, like previously we see when the softwares are not there, Disney has done all their jobs manually, all the studios they have done jobs manually. But after the softwares, the jobs became easy, the designs became easy. So might be in future, after 4 or 5 years, new design jobs and roles will be increased, and AI interface designers, the personalized designers or automation designers will be needed. UI UX can be improved, but the futuristic design should be also required the human
Hello.
intake. Yes, sir. Am I?
Yes, go to your results, sir.
Yeah. So what my conclusion is, I think you are asking about the conclusion, right?
Yes,
So, sir,
yes.
my conclusion is, what my thought is, AI will not gonna replace. AI is not a curse. AI is a boon for us. But in that case, we need all that design principle, all that basics which we have to learn. See, the why I am telling this because right now, see, some students are there when they will out and they will be in industry. They will probably, sir, why we learn all these things? AI is there and just I can type anything and I can found the result with generative AI or chat GPT bar.
So
or
this is
Google.
only review. You have done only review. Yes, review paper. This is only review paper.
Sir, it's not a review paper.
So can you show the experimental results or what methodology you have used?
Actually, sir, it's in my PPT, but I can't open my PPT here.
Okay, okay.
Due to some network congestion, that's why I'm showing this PDF, which I have submitted there. Sir, actually, I have taken data from students and artists. What the research I have done is, and I just go through with that. Okay, are you familiar with software or are you familiar with and techniques and all basics of the animation, basics of the design?
Okay,
So
Ankit, show your references.
Just give me one.
References are not in this file.
No, sir, it's there in this. Let me. So just give me 1 minute.
Hello, Abhishek.
Okay, Abhishek. If it is not available, then you can leave.
Sir, it's there. Everything actually everything was there in PPT, and I am just trying to find out that. But if sir, just give me 2 more minutes. Let me find that, or if I'm running out of time, then it's fine. I will mail it to you.
Okay, if PPT is working, then you can show the PPT or wherever the references are available. Okay.
Sure, sir.
Meanwhile, we are taking next presenter. Okay.
Thank you, sir. Thanks a lot. And I will just try to, if I will got it or it's working, I will try to that.
Yes, yes.
Okay, thank you, sir.
Okay, Abhishek, you can leave the presentation. Okay, I can. Okay, Pankaj sir.
Yes, yes.
Two
If any
scholars
other presenter is available, then we can
No, no,
continue.
sir, two scholars are facing technical issue. So
Okay,
the
okay,
session is over here.
Okay, okay,
So
sir.
we are very thankful to you. You have spare your valuable time. And
Okay, sir.
also
Thank you.
Sandeep Saxena, sir.
Okay, sir. Okay.
Okay, thank you
Thank
very
you,
much,
sir.
sir.
Thank you. Thank you, sir.
There is no other presentation, sir.
Yes, sir, no presenters.
Okay. Okay, sir.
Okay, sir. Thanks a lot.
Welcome.
Thank
sir.
you, sir.
Thank you, Pankaj sir. Thank you,
Thank you,
Sandeep
sir.
sir.
Thank you.
Can I
Yudhvir
close?
sir. Thank you, sir. Thank you, Pankaj sir.
Thank you,
Chitresh sir,
sir.
can I close?
Yes, yes,
Thank
sir.
you,
हाँ
sir.
yes yes yes.
Thank
It
you,
is
sir,
over.
for
चलो
listening
पंकज सर
to
आपको
us.
आपके दर्शन हो गए यहाँ पर.
Thank you, sir.
ठीक है चलो.
सर इतने
Thank you,
कुछ
sir.
नहीं हम
Thank
लोगों
you.
को
मिलते हैं
सुनने
फिर
के लिए
आपसे.
धन्यवाद फिर
मिलते
मिलते
हैं
हैं
सर, है ना?
ओके सर थैंक यू
And students, thank you very much. Participants, thank you very much for attending this session. I hope that you have learned a lot. There were only 2 presentations, and due to some technical issues, we could not have another 2 or 3 which were lined up. So extremely sorry for that. But these 2 presentations have added value to, and especially the presentation of Pratibha was really very good. Really very good. Thank you.
Thank you, sir.